Senior Research Fellow at ICGEB. She has focused her doctorate degree on drug discovery, and she has previously done a session with us on drug discovery and computational methods. Today, we will be having session 2 with Dr. Rini. Over to you.
Hi, good morning, everyone. Okay, so in my previous session, I informed about the structural-based discovery and etc. I started with Chimera basics, so I'll just carry forward on from that. Chimera on my system, so I'll just start with that. You just double-click on it. So whenever you open this, I hope it is visible, right?
Yes, it is.
Yeah. So whenever you open UCSF Chimera, on the right-hand side, you can see so many sessions, right? So these are the sessions which I've already worked on, and whenever you download it and you open a PDB or any kind of molecule on the UCSF Chimera, if you save the sessions when you close it and again open UCSF Chimera, so you'll get all these sessions on the right hand side. So you can actually directly use and check out these particular sessions. You don't have to go on open or fetch ID, etc. So I'll just click on this particular PDB ID. So now this is a folate biopathway enzyme, which I discussed earlier as well. These are 6 chains of this enzyme which have been crystallized, and the PDB has been submitted on UCSF PDB as such. So I'll just go and select one particular chain. And I guess I've already informed how to select chains and elements on this. So I'll just get on with it. So I'll just select chain A. So you can see that one particular chain has been selected. Now this is of the entire same enzyme. This is not different enzymes. Okay. So this chain has been selected. So I just need to work on this. So I'll just go for invert all models. All other chains has been selected. Apart from this particular chain, I'll just go and delete these because I don't need it right now. So you just go to action, atom bonds, and delete. Yeah. So this particular chain, if you want to focus this particular chain, you just go to this chain A, go to actions and focus. So we can see that there are few ligands or few atoms which are already visible. Now when you scroll on to these atoms as such, they will give you the name of the ligand as such. So this is HH2, that is pterin pyrophosphate. It is a intermediate substrate for this particular enzyme, and this does not have a drug right now bound to it, but you can always have a drug bound to it and then visualize it as well. So we can see that there's ATP as well over here. Yeah. Or rather you can go to select residue. All non-standard will be selected. APC is the one which is ATP in here. So it is an analog of ATP. So you can see here, and we can just go to actions and focus on this particular ATP. So see, right? So if you want to check the interactions and all, or if you want to see how many ligands or residues are interacting with it, so you can select either 3 angstrom or 4 angstrom data, which can be controlled from here. You just go to tools, go to general controls. A command line will come over here, and you just need to give this particular command, select ACP. So this is selected. Now, if you want to see how many residues are getting interacted with this ATP, you just give a space, write Z, less than 4. Now, less than 4 means less than 4 angstrom residues which are interacting with ATP will be marked over here. So you just select enter, and these are the ones which are interacting, which are under 4 angstrom of this particular ligand, right? I hope these are visible to you, which are in green color. So what I'll do, just for our visualization, I'll just color them in some other color, like take green, for example. So these are the residues which are under 4 angstrom of ATP ligand molecule. So I'll just select it by using control, upper arrow. This is selected. Now, if we need to know what heteroatoms are in this, we just need to go to atoms, color, and we can color it by heteroatoms. So here. So now we can see that this is the oxygen which is colored in red as such. The orange part is the one, this is actually phosphates. that is PPI attached to this particular atom, that is ATP. Okay, and these are the residues. So if you scroll over them, you can see which residues are these. That is cysteine, this is glutamine, isoleucine, aspartate, etc. So you just have a look at it whenever you are comfortable with this particular chimera. So arginine. So these are the ones which are binding, or these are under 4 angstrom of this ATP molecule. So we consider most of the time 4 angstrom and not 5 angstrom, because 5 angstroms covers quite many other residues which are not necessarily binding to the ligand. So if you want to see how many residues are that, I'll just remove 4 angstrom and I'll just write 5 on the command line and press enter. So we can see there are a few other residues which have been marked like this. That is tyrosine 268 is also marked. And we can just color it in different color like orange red. See. So this particular, these 2 atoms are also considered when you are selecting for 5 angstrom. And this is another ligand, that is pterin. Now this is the first substrate of this enzyme which attaches to ATP and forms pterin pyrophosphate, which I was talking about here. That is residue number HH2. Now HH2 is on the different domain of this enzyme. So what I'll do, I'll just go and focus on it. So we can see that this is the pterin over here. This is ATP, whereas this is pterin pyrophosphate. Now this is another substrate which is involved in folate biopathway of this particular enzyme. And we can see there's another ligand over here. These are the residues. And this is another ligand which is bound to this particular structure, that is PABA. So if you want to click on this, you just press Control arrow and it is selected, right? These small molecules are water molecules, and this is a metal ion, that is magnesium. Magnesium is required for the binding of ATP and PABA and all for this particular enzyme, and many of the enzymes need such metals so that they have a better binding to these natural substrate. Now you know how to select the residues and all. So if you want to see what is the difference between any of the 2 residues, what you can do, I'll consider PABA in this case. I will color it as yellow, okay, and color it by a heteroatom. And then I will go to the command line, add PABA, Z less than 4 angstrom. All these residues have been marked. So suppose if you want to see how far away is the oxygen atom of PABA and arginine NH1 molecule, so you need to go to tools, go to structure comparison or sequence. These are many things which you can scroll over and you can check what exactly they do. So you will go. So it's better to check how to first compare other molecules or other chains with this rather than going to the distance first. I'll go and give a brief introduction of how to do that. So what I'll do, I'll just go and fetch another ID which is of the same enzyme and I'll overlap it. So another ID for this is 5Z79. Now this enzyme, this enzyme is from Plasmodium falciparum. The 5Z79 ID is for Plasmodium vivax plus HPPK DHPS. So I'll just go and check X68WX. These PDB IDs can be extracted from PDBs from Google as such. So yeah. So if you want to see how many chains are in the other PDB ID which I have recently fetch, you just go to chain and you can see that there are 2 PDB IDs chain which are coming over. That is 5Z79 and 6HWX. For B, we can see that these are 2 here. And now C, D, E, F are the ones which are of 5Z79. So I'll just check on A, all. Go for focus. Now these 2 chains are different, of same enzyme from different organisms. I'll just go and invert all models so that remove the other IDs. So right now we just have these 2 chains of 2 enzymes from Plasmodium vivax and Plasmodium falciparum. I can just go to structure comparison and matchmaker. Now, matchmaker is the one tool in Chimera which actually overlaps the two chains of the enzymes, 2 or more than 2, in fact. So we can have around 500 chains also, which we need to overlap and check what are the structural differences among them. So I'll just check over here. I'll go to this reference structure. I will take it as 5Z79. The structures to match, these will come over here, and these are automatically selected. If you don't want to select this one, so you just go to this, you just give one click over here on this, but it's better to select them both. And you need to check if you want to change any of the parameters over here, then you can do it. If you want to see that best aligning pair of chains, this is the default one. It's better to go for this. Or if you want to do specific chain in reference structure, the chains which I have deleted right now, like C, D, E, F of 5Z79 or B of 6JWXPDBID, you could have done it from here also. So what would have come on the reference chain is that all the chains would have come over here and the structures to match as well. So you don't have to delete it also, but it's better to delete it so that there'll be too much chaos on the Chimera session or else. So you just go and apply. Yeah. You just click select. Okay. Yeah. So we can see that these are totally overlapped on each other, right? There are a few structural differences, but since these are same enzymes from different species, there's not much difference. However, the most striking difference which I can see over here is this one, that there is an extended alpha helix in 5Z79, number 0 chain. That is, you can see and check it from here, that is 5Z79 is number 0, and 6JWX, that is in pink one, is number 2. So when you scroll over here, we can see that there is an extended alpha helix over here rather than 6JWX has a loop over here, right? And we can see the active site. The active site being this one and this one. So I'll just go and select the residues. Take PABA, for example. It is selected over here. And all these residues which are bound to this particular active site is shown over here. Now, if you want to see the surface of the entire structure, what you can do, you just need to clear selection first. Select all, go to surface, go to actions, then go to surface and show. Yeah. So now this is the surface view of both the enzymes which are colored differently, and this is the active site which we have earlier marked in red color. That was the active site of pterin binding site. This one is here and another one is here. Since this is a surface structure, what you can do if you want to check where exactly the ligands are binding, you can go to surface, go to transparency, and you can do it for 90%. So we can see that there is surface also, and I'll just change the color of the background. You just go to color, all options, go to background and mark it as white. So for the visibility. Now, since the surface is 90% transparent and we can see our PABA or another active site over here, all the substrates are bound to here and another active site is over here. So now if you want to see how many residues are bound over here, we can go for the same thing, that is select PABA, Z less than 4, all the residues will be marked, and we can just change the color of that. Right, so this is the basics, like, structure alignment, and you can also do sequence alignment from the same tool, that is go to same tools. Structure comparison is the one which will compare all the structures for this. Matchmaker is the one which will actually overlap all the chains, whatever you are selecting. And match and align is structure-based sequence alignment. So what will happen that whatever the structure is there, the sequence for the same will be aligned as per the structure and not as per cluster W acid. So you just click OK. Yeah. So this window will pop up, and now this is the sequence alignment which we can see over here. If you want to check, suppose take, for example, this particular residues. Where exactly are they lying? So I'll just select them on here, and it will be selected over here as well. So I'll just go and focus. Move the surface of the same. Yeah. So the structure, the sequence which I have selected, I'll select this, go to this, and color orange red. So we can see that this particular alpha helix, the residues which I selected from the structure sequence alignment are shown over here. So if you want to see where our conserved residues are also marked highly, what you can do sequence alignment from cluster W or omega from Google, and then check it on the structure of the from UCSF Chimera. So yeah. So do you have any questions if you want to check apart from this, anything else? Now, if you want, So if you want to remove these atoms, you just need to select this particular atom, upward arrow, and you can go for atom bonds and hide. Or you can also hide other residues by going to the command line, selecting PABA, Z less than 4, and you can hide the structures if you are not interested in looking at those. So these will go. And you can also remove the ribbons as well, right? So it's just playing around with Chimera, nothing else. Now, if you're interested in looking the distances between two residues, you just go to Tools, General Control, Command Line will also be there. There's a strip for distance as well. So, but it's better to do it from the viewing controls and all. So you just go to structure analysis. So what you can do, you can click on distances. One window will pop up. And now you need to check which two atoms you want to consider and which two atoms you need to check. So you just select all, focus. And if I want to go for PABA, for example, or pterin, you just select this particular, this is CKL, this is a drug bound to 6JWX PDB ID, or you just select over here. Yeah. This is PABA, you just select control, select entire thing. And if you want to know what is the distance between PABA's nitrogen atom and serine 382. So you need to select this particular. You just select control, select, and control and shift, select the serine residue, simple. And then click on the structure, measurements, and create. So we can see over here that there's a difference of around 3.418 angstrom between PABA's oxygen atom and serine's nitrogen atom. Serine's oxygen atom and PABA's nitrogen atom. So I'll just color it with different color, like red, so you can see. I hope it is visible, 3.418 angstrom. Or else you can also check on the structure measurement, the distances between two atoms as such. You can also go for angles and torsions and adjust torsions as well. So there are many parameters in there, right? So I guess this is enough for now, because all other things you need to check once, and you need to understand how to play around with UCSF Chimera, and you need to know what exactly you want to do. So all this, if you want to make a figure for that Chimera is the best tool for high resolution images. So if you want to click on this particular area and you want to show this figure as such, so what you'll do, just go to File, Save Image. So it will give another folder which you can always change from here. You just give some other name. And to have high resolution figures, it is better to go for pixels, and image width you can change to like 10,000 or 14,000. It doesn't matter. The resolution of the figure should be very high so that a person who is looking at the figures can understand what exactly is going and what exactly are the differences between two figures. So I'll just write it out as 14 and do a tab. It will show accordingly maintain the current aspect ratio and save it. It will take a few seconds. Yeah. So now if you want to check how the figure was saved, you just go to that particular folder and check it once. Yeah. So this is the figure which I just recently saved. And as you can see, this is a highly high resolution figure, and you don't need to change any resolutions in the figure as well. So I guess this is enough for Chimera. As of now, if you have any problems in Chimera, you can always contact me on my email ID, or if you want my phone number, then yes, definitely you can take it from Michaela as well. And so I guess I'll start with Schrodinger now, if it's okay.
Yes, that would be good.
Okay. So Schrodinger is, before starting Schrodinger, actually, I just need to show you one particular slide. Yeah. So I hope this is visible, right?
Yes, you had a PPT, Shai?
Yeah.
Yes, it is.
Okay. So whenever you are looking for a drug which is a target of a particular protein, or if you want to target a particular protein, it's always good to know what exactly you want to do and how you want to proceed with it. So my protein of interest are HPPK, DHPS, and you can do in silico screening also, and then you can dock the structure onto the particular protein's active site, or you can do blind docking. There are many parameters and there are many factors which you should consider, and it's totally onto your work what you want to do exactly. So it relies on that. Basically. So if you want to go for in silico screening, there are hundreds of chemical databases now, which you can, from where you can check your drugs as such. So few of the chemical databases I have mentioned over here are NCI, that is National Cancer Institute drug database. These drugs are basically used for cancers, different types of cancer, and whatever a researcher has checked a drug, what they do, they check the drug, and whatever kinetics or whatever factors they have considered, they upload it onto this particular database. Another database is Drug Bank. Now, Drug Bank is the one database which has all the FDA-approved drugs. So it is always really good to check from the Drug Bank database, because the drug is already an FDA-approved drug. And if that drug gives you a good binding affinity with your protein, then it's a really good thing to do. It's really great for you that you found a drug which can actually bound to your protein, and it is showing any kind of inhibition as well. And it's already FDA approved, so you don't have to, take so much stress that what would be their toxicity levels and all, because all the details are present in this Drug Bank as such. Another chemical database is Zinc. Then PubChem has the largest library of all the chemical compounds. MMV is Medicine for Malaria Ventures. They also have thousands to millions of compounds which you can test on any kind of protein. We have Pathogen Box. We also have COVID Box now. So you can check for the compounds for SARS-CoV-2 as well. And those compounds can also be tested for another viruses as well. These are some of the chemical database which I am telling you right now. There's another chemical database for Schrodinger, which Schrodinger has and patented for their own use as such. So you can go and check for in silico screening from these databases. When we are talking about docking and searching for a chemical database or a compound which you need to check for, there are 4 to 5 things which you need to consider. Whether you want to go for structure-based drug discovery or ligand-based drug discovery, either fragment-based drug discovery or machine learning as well. When we say structure-based drug discovery, what happens is that you need to consider one particular active site or the entire protein, so that you can check its structure, which I was just telling you on the Chimera, the active sites for the proteins. And then you can actually click on those residues and check whether the particular drug which you have in mind will bind to it and give good docking scores and high energy as well. when we talk about ligand-based drug discovery, the ligands, we can take one particular ligand and we can find multiple derivatives for that and or analogs as well. So if you have particular, suppose this is, take ATP for example, if you want to find an analog of ATP and you need to check whether it is binding to your particular protein of interest. So you just take it and then put it onto the parameters from various drug discovery platforms like Schrodinger, BioVia. There's another program for that, Autovina, etc. Another is fragment-based drug discovery and machine learning. I won't be focusing on machine learning because I don't know and I haven't used it as of now. So I'll be basically focusing on structure-based drug discovery and ligand-based drug discovery using Schrodinger. So if you need to download Schrodinger, you just go to Google, type Schrodinger. It's always better to have a Schrodinger license. If you have it, then it's really good because you can use all the platform from that. And you just click on this particular site. You go to platform and go to download software. You need to sign up for this first. I already have it. And once you sign it, I'll just write it in front. So once you sign it, you will have this type of window. And from here, you can actually change the version as well. Depends on your computer's latest version. So I will be taking this release 2022 to 1 because my computer is quite the latest one. And you can choose your OS. It can be Linux, Windows, Mac. I have Mac, and you can just go for download it. I already have this, so I will be focusing on the Schrodinger. So when you download Schrodinger's Maestro version, it will look somewhat like this. Yeah. Okay, so this is the Maestro's window. And this is a module for Schrodinger from where you can do docking studies, you can do energy landscape studies, you can also go for simulations of a protein or ligand, a protein docking, etc. So when this particular window opens, first of all, it's really important to know that whatever you are working on, it needs to be saved. If you're not saving it, then you will lose all the data. So you just go to File and go for New Project. So wherever you want to check, you can file this or you can save it, and it will be saved in maestroproject.prj, and you just save it. Okay. So now you also need to change the directory for that, change working directory, and I will take it into consideration, this one only, and I'll just click on test PRJ and choose. So whatever work I'll be doing on from now onwards, this will directly be saved on this particular directory. So just like Chimera, if you are working on a protein, you need to fetch the ID, or if you have a homology modeled structure, then you can do it from here. So what I can do, I'll just go to this protein preparation. I'll click on that. Now it works in this manner, right? So you just go to get PDB. So you can check any PDB, take 6JWX for example, and you will just click on download. So here we can see the 2 chains of 6JWX are here. The controls are somewhat similar to Chimera only when you are using your mouse. I have not shown the ribbons if you want to check the ribbons. In Schrodinger, what happens is that all the ligands, proteins, solvents, and other molecules come over here on the workspace. Now, this is the workspace, and this is a table from where you can check what entries has been done. So you just click on table for once, and you can see whatsoever entries have been made. So you want to check what exactly is this. So this is crystal structure of Plasmodium falciparum HPPK DHPS wild type enzyme. The PDB ID is this. The PDB resolution is around 2.5 angstrom. This was done through X-ray diffraction. And all these parameters you can check from here by scrolling left and right. Okay. So it's here. So if you want to change the protein and you want to look at the ribbons or anything like that, you just go to presets and apply custom preset. So we can see all the atoms and all the ribbons. Their labels are also turned on. So if you want to check on tyrosine 132, you can just click on it and you can see so many things in here, right? So if you're not interested in looking at the labels, so you just go on the right panel and just click on annotations toggle. So all the labels will be moved. So if you want to see what is the interaction going on in this particular enzyme, so you can just go for ligand interaction. Now this gives a very nice figure for this, and we can see what interactions are actually going on. So this is CKL. This is a drug. I'll just go on PH2. This is the pterin substrate which I was telling in Chimera session, the first substrate of HPPK DHPS. And we can see that this pterin is having interactions with tyrosine, phenylalanine, asparagine, arginine, serine, etc. Now this green color shows over here that it is showing a pi-pi stacking interaction with the center of this particular pterin ring. Similarly, since tyrosine and phenylalanine are aromatic amino acids, they always do pi-pi stacking most of the time. And this particular green, orange, and this color, it shows the interaction level of different things. Like we can see that valine, proline, glutamine, and tyrosine, they are having hydrophobic interactions, whereas the red thing shows negative interaction since arginine is over here. And these are the water molecules. You can check it over here. And these amino acids are also colored as per their solvent or hydrophobicity, etc. So if you want to check hydrogen bonds, you can select over here. Salt bridges can also be done. Waters can also be removed. Protein, water, hydrogen bonds can also be done. So if you don't want to see pi-pi cation, it will be removed. Yeah. And if you want to check and save it, you can save it here. You can remove the LID legend, which was over here, and you can save the image from there. So this will be a transparent background, and wherever you want to check it, you just change the name and save it. So you will find it. In whatever folder you have done, okay? So this is ligand interaction. If you want to go and check another ATP or any other ligand interactions as such, so you just click on ligands over here, right hand side, and you can see the interactions. So you can see that magnesium is having any solvent exposure with this particular ATP molecule, and we can see that histidine is making pi-pi stacking and phenylalanine along with it. So you can always check all the kind of interactions which are happening. Now this is a drug which is bound to this particular enzyme, and we can see this particular drug, that is a sulfur-based drug. These are the level of interactions which is happening when bound to the active site of the enzyme. So we can see there is pi-cation bonding, there is pi-pi stacking, there is polar bonding, etc. Right. So apart from this, it's really important whenever you run a job, you need to know where the job is running and what is exactly going on right now and what jobs are running. So you just go on the right panel, click jobs, go to monitor. So this particular window will open up, and you can see that from this project I have recently saved. So no jobs are here right now because I have not run any jobs. But you want to see how many active jobs are there. Right now, there's this active job is running, that is induced with docking, and you can check at what level it is. You can just scroll down and up. You can go to the details of this. So this is the output file of this. It's still running, so I will not touch it right now. And you can click on all the jobs which have been run till now. There's only one job which has been run till now, so it is still running, so I won't touch it. Yeah. So apart from that, if you want to check where your ligand is on your keyboard, just type L. So we can see when you scroll over it, the name of the molecule comes on the lower panel over here. So you just check that is this is AMP. It is showing interactions with different residues, which we also saw on the 2D label. So it is showing pi cation, pi stacking with this particular ATP molecule. So if you're not interested in looking at the interactions, you can just go to the right-hand side lower panel, click on it, and you can see all these non-covalent bonds, pi interaction and contracts as such. So we have just selected bad and ugly ones. If you show the good contacts, these are the many contacts which are getting involved in this ATP binding. So you just remove it. And if you are not interested in hydrogen bonds, you can remove it from here. You can remove halogen bonding. You can remove pi stacking. You can remove pi cation as such, salt bridges, and you can also see aromatic hydrogen bond. So it's totally dependent on you what interactions you are interested in. And we can change the background of this particular window by using the workspace toggles. And I'll just click on black for now. So this can be changed over here. And we can check it in tile as well. If there's another enzyme molecule which we have selected, that will come over here. So I'll just do it as simple tile entry. Okay. So moving on to protein preparation, it's really, whenever you are doing any kind of protein study, it's really important to understand that the protein should be in its minimized structure. So protein preparation wizard or workflow of this particular Schrodinger is that it's made in such a manner that it will utilize OPLS force field. Now this OPLS force field is the one which has all the parameters required for minimizing the protein structure so that it is in the minimized state. And whenever a drug wants to bind to it, the protein is not in highly movement so that the drug couldn't bind it. So it's really important that protein should be in minimized manner, and the drug should also be in minimized structure at that time when you are doing any kind of docking studies. So we have taken get PDB from here. We can pre-post process it. It will take 5 to 10 minutes. So you just click on this and you can either align it as according to Chimera as well, align to any kind of first selected entries, or you just click PDB ID. You can give any other PDB ID and select on it. So it will align to that particular PDB ID. So we have already done that in Chimera, so I won't be repeating it. Now these are the bond orders. Whenever a structure is submitted to RCXP PDB, there is a possibility due to its resolution or any kind of other parameters, few of the side chains or few of the loops are missing, or they are either disordered. So what Schrodinger does is that it can actually fill in the missing loops using a module of Schrodinger that is prime. And if you click on it, whatsoever loops are disordered here, they will come and they will, it will give various energy structures for all the loops. It will give around 10 to 12 loop structures, and it's better to select the best loop structure with least energy so that it binds to it and it gives the structure a full-fledged structure as such. And if you want to delete what is beyond 8 angstrom, you can delete it, or it's better to delete it till 5 angstrom. So these water molecules, we do not need it beyond 4 or 5 angstrom, because 4 angstrom is the one where active sites have all the waters and all the ligands are there, and we are only interested in that particular angstrom site. So I'll just move it over here. And you can also change the heterologous state with EPIC. This is also another module which changes the protein molecule structure using pH. So if suppose your pH is around 7, so you can do plus minus 2. It's right now 7.4. So if I'm not interested in that, I can move it. And if suppose my protein works perfectly in vitro experiments at around 8 pH. So what I'll do, I'll just click it at 0.9 or 0.8 or 0.5. So this will give at pH, and it will minimize the structure as per in vitro models only. Right. So after pre-processing, what we can do, we can optimize the hydrogen bond assignments. We will just go to settings, and it will change the water orientations and fit all the water molecules in such a way that the water molecules are not coinciding, or they are not interacting, and they are not coinciding with the ligands as such. So we will click on this, and if you want to use the crystal structure, it's totally up to you. And since we have already minimized hydrogens of the species earlier in pre-processing, so if you have any kind of mutations or any kind of altered species, you can click on this particular thing as well, and then optimize it. And cleanup means minimization of the protein, so you'll just click on it, and all the heavy atoms can be converged from 0.3 angstrom to, if you want to increase the angstrom, you can go till 1 angstrom. It's suggested not to go beyond 1 angstrom. And you can optimize hydrogens only also as well, but it's better to optimize the entire structure. And we can delete waters from here, from ligands, near the ligands at around 5 angstrom or less, and then you just run it. So that will take around 10 to 15 minutes. So it's better not to run it right away, because it will take quite a lot of time. So if you are interested in particular one chain, you just go to chain protein, click on chain B. Now this entire structure is selected. Right click on it and delete atoms. Ligands can be deleted, CKL of chain B and AMP of chain B. Right? If you are not interested in any kind of water molecules also, you can just click on waters, and all waters can be deleted. Right? So, Michaela, how much time is left?
We have around 15-20 minutes.
15-20 minutes. Okay, so I'll just go and tell you about the docking as such. So, when you are talking about Schrodinger, they have different modules and they have different tools. So you can check on go to tasks and click on browse. So for docking, we use Glide. So you click on Glide, and these are the ones which we can check. So whenever we are docking, we need to understand and we need to give the computer a program that where exactly we want to dock onto our structures. So for that, a grid should be generated. So I'll just click on this receptor grid generation. So you need to understand, if suppose this particular site is the one. Yeah. So suppose you are interested in docking all your ligands over here, this particular site. So what I'll do, I can do it in 2 ways or 3 ways. I can do it in the Centroid or Workspace ligand, which can be done from here. We can click on this to pick and identify the ligand, and I'll click on this particular ligand. So this box comes, and it is actually taking all the residues around this particular ligand, and it shows that this is the active site from on which our ligand should be bound. So I can do it in this manner, or I'll just go to site, click on Centroid of selected residues, and I can specify it over here. So whatever number I want, I can select it over here. Take 381, 382, 384, 434, 481, 561. I'm just taking random residues as of now. So these residues I have taken into consideration as of now. I'll just go on add and then click OK. So these residues are the ones which will define the active site of the molecule. I'll just click on it, OK. And these are the ones, this is the site which I have selected. The residues, whatsoever residues I gave, it's around here. So whatever ligands you want, which have been pre-processed, they will come and bind onto this. Okay. So you can do it by this, or you can also go for XYZ coordinates. Whatever XYZ coordinates you want, you can select it from here. Take 6.231. Y coordinate should be around 35.2. And you can see like 17.12. So when we have given the XYZ coordinates, the active site, this particular glide box, comes over here, and we can dock onto our structures on this particular active site. So whatever ligands we give for binding, it will come and bind to over here. As of now, I know that I want to bind my structure on this particular ligand. So I'll just click on this and click on the ligand. So the box will move accordingly. Now you can also do one thing. You can also go for blind docking on the entire protein structure. So for that, it's better to not click on a ligand, go to site and give your XYZ coordinates of the protein. So take 17, this is going up. So you need to play it so that the entire structure is taken into consideration. You can change the numbers and you can fit this particular box, this particular enzyme in this box. So you need to take like this, and you can change this as well. 1.58 minus 78, 7.567. Okay. So this is going right now outside. So you need to shift this particular box and fit this enzyme in the box. So you need to check all the XYZ coordinates and play with it. So you go to this. Yeah. So all other parameters should be considered as default only. So if you want to check any groups which are rotatable and you don't want them to rotate as such, like phenylalanine, tyrosine, etc., you can select it from here and then you can deselect it as well. So what I'll do, I'll just go for it. I'll just do it, change the name for this, and on job settings, you can actually select the name of this, and directory for grid files also can be done on this. If I have selected already and if I have a file which I want to load onto this can be done over here. So I'll just go and run it. Now job is submitted and we can check its, we can monitor it over here. Go to monitor and we can see that it died for some reason because all the CPUs are running on for this particular job, right? So it's just like this only. And so when this job is completed, I can show you another one, but it's actually busy right now, so I don't want to, use it as of now. Apart from that, what you can do, you can actually take all the ligand structures which you have downloaded from PDB IDs and all others. You can go to Google Chrome. You can check drug bank. Now this is the database which I was telling about, FDA approved drugs, and you check on any molecule like, take 0141. So this is N-acetylglucosamine, and this is a small molecule. It is already in use. It is approved, and all the details for this particular drug will come over here. So what you can do, you can either download it as .mol or .stf. I'll just download it as mol. I'll check and save it in notebook. Yeah, this can be saved on Word as well. And then you'll upload it on Schrodinger. So what I'll do, I'll import structures which I have already, and I'll go on another project of mine. So I will take this particular ligand library, which I have already prepared, and here. So these drugs have been prepared as of now, but if you want to check, you can always import it from here, import structures, and all the drugs which are not processed will come over here. And then you can go to tasks. You can either write it or browse it over here. You can go for lig prep. And then you can use structures from file, or you can use structures from project table, which is over here. All the structures which have been selected are in the project table, and in the workspace there's only one as of now. If you want to use all the structures in the workspace, you just click on Shift and select on these points. So you can see these are all the ligands which have been pre-processed and not yet docked onto the structure, right? So you go to this and you can go for lig prep. Now project tables, it will also give the number of selected entries, and in Workspace also now we have 128 selected entries. If you are not interested in taking Workspace things, so you just clicked on this. So you will check. So we can see that in project table there are 128 selected entries which are shown over here. Okay, or you can take one Workspace entry. This is just one, and all these will not be selected because we have taken only one Workspace entry, right? So these are all the default parameters. If you want to generate at most 32 ligands, you can do this. It's better to go for generate all combinations rather than going for all these things. And the output format for the same will be either Maestro or STF. So it's better to go for Maestro because this is Maestro, and you can also get output in STF file as well. So you just go on this and prep text and then run it. So for preparing ligand, it will take around 5 to 10 minutes, not more than that. And the output format is this one only, which I have already selected, which I already have it. So that's why I'm not selecting it as of now. And I'll select it. Now these are the ligands which are already prepared. And these ligands are ready to be docked onto the structure for the grid generation, which we recently did. So we'll just go on this. And I'll just go and import the structure. I'll just go and go for ligand. So over here, when you write docking onto this particular thing, we can see that there are different parameters and different tools for this. So you can also do peptide docking, you can do protein-protein docking, you can do covalent docking, you can do ligand docking, you can also do induced fit docking and one-step ligand docking. For this, we'll be using the normal ligand docking. So this windows open up, and receptor grid generation, the box which we showed for glide generation, we can take it from here and browse the file wherever the grid has been prepared. So I can take it from another module. Yeah. So you can take this. So this will be a zip file. We'll open it up. So this is loaded onto this particular ligand docking, and then you can take the ligands from either file or project table, which are already selected right now. You can go to settings. Now there are different modules in this. What you can do, if you have very large number of ligands, then you should always go for high throughput virtual screening. But since the number of ligands are not that much over here, it's just 128, it's better to go for either standard precision or extra precision. Now standard precision is the one in which ligands will be docked onto very gently, and the docking score will not be the exact one. So when we are talking about extra precision, if we have lower number of drugs, you should always go for extra precision. Extra precision means that the software will take the ligand molecules and dock the structure of the ligand onto the receptor, that is a protein, in its active site highly precisely. So we can check all these things. If you want the ligand to be highly rigid, then you can do it like this, but it's better to take it as a flexible molecule only. And we can select and deselect all these things if you are not interested in this. And all these are default parameters, so it's better to take it into consideration in that manner only. And you can also go for advanced settings and check what exactly is going on. And if you want to include aromatic hydrogen as donors or etc., all these you can play around, and it's totally dependent on your work, what you exactly want to do. So you just go and change the name of the file you want to do, and then go for run. So this will take around 2 to 3 hours for docking as such. So I can show you the docked structures, which I already have. So you go to import structures, and I have this one. Yeah. So the output file is out.megz. We can go to this. Yeah. So all the structures are come over here. Right? So the protein structure which was monomerized is selected over here. Now, if you want to see, suppose take for example this particular compound, you select it using shift, and we can change the structure like this. We can see that this is the binding site of this particular protein. And if we need to know where the docking scores are, we need to go to table, and we can check the docking scores on this particular panel. Or we can go to search. You can just go for this. We go to glide, primary. First we'll remove it. We just need to check the docking scores as of now. So I'll click on docking scores, glide E-model, XP score, and all these. We are just interested in docking scores, so we can see that the J1D compound and the 6JWX compound has a docking score of -10. The docking score should be, it's always preferred that the docking score should be more than -7. So -10 shows that it is a very good compound. It is binding highly efficiently in the structure as such, right? And now you can see its interactions. I'll just go for custom preset. So you can see over here that our ligand is here, and we can change the color of the ligand for our understanding. So if you're interested in removing the hydrogen molecules, you can do it like all hydrogen molecules will be removed. Or if you want to see all the hydrogen molecules in this, you can see it like this, but it's always better to remove it because it's way too much cluttered. So we can see that the structure is docked onto the receptor, that is our protein, highly efficiently, and it is going inside the active site. Right. Now, if you want to see the interaction with this, you click on this, and you can go for this. These are the good interactions. We can see that the hydrogen bonding is the yellow one, which we can see these dashed lines here. So you just click on this and you can go to expand and the 4 angstrom. Click on the 4 angstrom. All the residues which are in proximity to this particular ligand under 4 angstrom will be selected, and you can actually show it in this manner.
मैंने जाना कहाँ गया धो लो मैं नाश्ता करके जाऊंगी नहाने.
So we can see that these residues are the ones which are selected. Or you can go to ligand interaction diagram and we can see its interaction over here. This is the unknown molecule. So yeah. So we can see that it has high docking score and it is showing many interactions. So we can not only rely on the docking scores, we need to also check its interaction, how many interactions it is making, and what is exactly the energy of this molecule. For that, for finding out the energy, we can always go for MMGBSA scores. It gives the Gibbs free energy, and if the Gibbs free energy is less than -50, then it is considered a very good compound. For doing MMGBSA, we need around 1 or 2 days, because if we run the job right now, it will be completed by tomorrow. It takes quite a lot of CPUs as such. And we can see over here that arginine, phenylalanine, asparagine, they are making multiple interactions with this compound. So this compound can be considered a very good compound as such. So if you are interested in simulation studies also, we can carry on with this, but it's better to check the MMGBSA scores, so I can tell about the MMGBSA scores and simulations in the next session as such. But as of now, you need to understand what exactly is going on, and there are different poses of one single compound only. So what we can do, I'll just select on this another compound, the same compound but different pose. And I can merge these three. Yeah. So we can see that the pose of this particular, the J1D, another pose of the same compound, is quite different from the reference molecule. So we can check what is the docking score of the another one. The first one was around -10, and this is around -9.957. So this is also docked onto it beautifully. And you can compare the interactions as well of this. So just go to ligand interactions. And this is the one. Yeah. So the interactions are also good. The docking score is also good. But we need to understand its energy. So we can do the MMGBSA score calculation in next session. And the simulation studies can also be done in next session. Because simulation takes quite at least 1 week or so, depends on the CPUs or the jobs which are already going on your computer. So it will take quite a lot of time. What I'll do, I'll run all the jobs and I can show it to you in the other session and save it for your own convenience. And you can always save these things, like save image and all, for the new drugs.
That will definitely be very helpful, Dr. Rini. Dr. Rini, one question. What is the difference between binding free energy, docking score, and the binding affinity?
See, binding affinity, free scores, and docking scores is like we have a ligand, and the software will tell the protein to just go and fit into that particular active site. Binding free energies is like, suppose this is a receptor, this is a ligand, and it is binding onto it, but it is moving quite a lot. Right, so the free energy is not very low. So if it keeps on moving now, what will happen after some time? It might get out of the receptor as well. So free energies should be as low as possible. As we go on the formula, delta G equals to delta, what was that formula? Delta S minus T delta H, I guess. delta H minus T delta S, yeah. So the Gibbs free energy should be as low as possible. So this is the major difference between the two.
Okay. So thank you so much for such a comprehensive hands-on training on Schrödinger and Chimera.
Thank you.
And are we winding up for today?
Yeah, I guess that's enough because most of my jobs are running right now, so I cannot actually.
Yes, we can see that.
Yeah.
So thank you so much for such a comprehensive hands-on experience with Chimera. And as we all can see that there is so much more to learn, and we will be adding more posts on our social media regarding the next session with Dr. Rini and the topics that will be included in session number 3.
Okay.
Thank you for joining us today, Dr. Rini, and we look forward to learning more from you in the next sessions.
Thank you.
Thank you so much. Have a good Sunday.
You too. Bye.
Bye-bye.